So welcome back friends. This is online lecture of biotechnology. Myself Dr. Sharad and I am going to discuss you on the topic Introduction to Fermentation, in which we'll study types of fermentation process. So already we had discussed batch fermentation process. Now our next topic is with batch fermentation process. So already we had discussed regarding bioreactor, types of bioreactor, and the ideal parameters required for designing of bioreactor. Then how lab scale level fermenter look likes, as well as industrial fermenter look likes. Now we'll see in detail about fed batch fermentation process. So in batch fermentation process, it is a modification of batch fermentation process. We add everything, right? And we are giving a sufficient time. So after giving sufficient time, we'll get product, and we take out that product and go for downstream processing. But in feed batch fermentation process, we supply the substrate in small amount. I'm repeat, this is very important. So in feed batch fermentation process, we feed the substrate, we add the nutrient source in small amount throughout the time for the cultivation of various bioproducts, right? So these are the various diagrams of fermenter in which various fermentation processes are carried out. For example, this is batch fermentation process, this is the continuous fermentation process, and as well as batch fermentation process. So we are interested in this feed batch fermentation process. So it is obviously a modification version of batch fermentation process, and we are adding the substrate in a different time interval, of course, for fermentation, and we keep that substrate prolonged log and stationary phase. And here important thing is what we could not observe in batch fermentation process. In batch fermentation process, we'll get a product, same in case of feed batch fermentation process, but the form of product. In batch fermentation process, for example, we get alcohol from carbohydrate, but in this feed batch fermentation process, our requirement is what? The biomass of uh, organism. So we require, we will take an example of yeast, Saccharomyces cerevisiae, which is a baker's yeast. So if our target is to get the baker's yeast, means we require the biomass. So this fermentation, feed batch fermentation, results in the rapid increase of biomass. So, and thus the feed batch technique is an improved version of fermentation. So by avoiding the disadvantage of batch and continuous fermentation, we go with feed batch fermentation process, right? So in batch we add everything, already we have discussed. We are giving sufficient time, and then we'll get the product and go for downstream processing. In feed batch, what we do, we add a substrate in small amount in different time interval, and we'll get a product. We'll take one example of glucose. Sorry. Say, suppose we have a 10-liter carbohydrate, say example glucose, and we add organism, we wait for, we keep this fermentation for 7 days, and we'll get product, for example alcohol. This is the example of batch fermentation process. But what exactly the difference observed in feed batch fermentation process? If we take same capacity of bioreactor, for example 10-liter, so we add once we add the substrate in installment. So in first case we add 1 liter glucose, then after time interval specific we add another small amount, for example 3 liter glucose, and then after some time interval we add 6 liters. So ultimately our the capacity is fulfilled. So 10 liter is glucose already added, but the time interval is different. Now question is that why we add substrate in different time interval? right? The answer is that there are some substrate, they act as a toxic for bacteria. When there are some substrate, they act as a toxic for bacteria when we add this substrate in a bulk amount, or we can say in a once. At a time, if we add the substrate, then it will be toxic for production of specific product. So such cases we add small amount of substrate in a different time interval. That was the basic difference between batch and feed batch fermentation process. So why that effect of substrate on that bacteria when we add in a high amount? The reason is that, for example, your substrate is glucose, and if we want baker's yeast, when we add substrate in a once, then the viscosity increases, and the viscosity increases, then definitely the oxygen mixing is less into the fermenter, and hence the organisms are not survive, and it will be toxic to definitely bacteria. At the same time, they repress the fermentation process. That was the case which was not observed in batch fermentation, but it is observed in feed batch fermentation process. This is the basic difference between batch and fermentation process. Take an example of production of baker's yeast. So here what our requirement is, what we want, biomass. So increasing biomass. So our byproduct is, say for example, alcohol and some other byproduct is there, but our main product is biomass of our organism. So in such cases we use feed batch fermentation process. So our desired product is yeast. So for gaining the biomass production we use feed batch fermentation process. But for alcohol fermentation process we use batch fermentation. It is good type of fermentation for alcohol production. And for yeast biomass production we use feed batch fermentation process. So this is regarding feed batch fermentation process. So we will see in next lecture regarding continuous fermentation process. Thank you.